So good afternoon, everybody. Silence, silence. So we'll start. Rapoport-Luebering Cycle. So in case of glycolysis, what we have seen? It starts with phosphorylation of glucose. What is the first molecule formed in glycolysis? Glucose, then glucose, then glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6 bisphosphate, then dihydroxy acetone and glyceraldehyde 3-phosphate. Here come glyceraldehyde 3-phosphate. Then dihydroxy acetone phosphate, interconvertible with the glyceraldehyde 3-phosphate. So as glycolysis continues with glyceraldehyde 3-phosphate, dihydroxy acetone phosphate is converted into glyceraldehyde 3-phosphate. From here, two molecules of glyceraldehyde 3-phosphate will be produced. From there, 3-phosphoglycerate, from there 2-phosphoglycerate, phosphoenol pyruvate, and then pyruvate. So pyruvate is the product of anaerobic glycolysis. If it is, pyruvate is the aerobic end product of aerobic glycolysis, and if it is anaerobic glycolysis, then lactate is the dead end product of glycolysis. Now, in Rapoport-Luebering cycle, this is nothing but a shunt pathway of glycolysis. Okay? It occurs in mature erythrocyte. About 1/4 of the total glucose metabolized in the RBC are utilized in this pathway, Rapoport-Luebering cycle. It is concerned with the formation of 2,3-BPZ, bisphosphoglycerate. You already know what is 2,3-BPZ. What is the role of 2,3-BPZ in hemoglobin, oxygen dissociation. So here you see in glyceraldehyde 3-phosphate, it is converted to 1,3-bisphosphoglycerate. One step is here, 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate, it is converted into 3-phosphoglycerate in the glycolysis. But in case of Rapoport-Luebering cycle, this step is not taking place. It is taking another pathway. This 1,3-bisphosphoglycerate, it is converted into 2,3-bisphosphoglycerate by 2,3-bisphosphoglycerate mutase. So this mutase enzyme, it shifts the phosphate group from carbon number 1 to carbon number 2. So this 2,3-bisphosphoglycerate is produced. Now how this 2,3-bisphosphoglycerate is converted into 3-phosphoglycerate? By the enzyme 2,3-phosphoglycerate phosphatase. Phosphatase is the enzyme which cleaves the phosphate group from the substrate. So again, from 2,3-bisphosphoglycerate, 3-phosphoglycerate is produced, and further the rest of the glycolytic cycle is followed. So here in the glycolysis, we get one ATP. This is catalyzed by the phosphofructokinase, glycerophosphate kinase, phosphoglycerate kinase. This is catalyzed by phosphoglycerate kinase in case of glycolysis. Hello, hello. Why you are gossiping? So this part is not taking place in case of Rapoport-Luebering cycle. So we are losing one ATP here. So in case of Rapoport-Luebering cycle, what is the energetics? There is zero production of ATP in case of Rapoport-Luebering cycle. So what is the importance of this cycle for taking in the RBC? What is the importance? The importance of this cycle is to produce 2,3-BPZ, which is required for the RBC or hemoglobin when the partial pressure of oxygen is very less in the air or in the blood. So one thing I would like to tell you, in case of this this 1,3-bisphosphoglycerate, this glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate formation, there is a NADH production. So in this case, why energy generation is zero? This NADH can be oxidized in the electron transport chain. We can get ATP there. Why it is not taking place? Because, can you assume why it is? It is because it is occurring in the RBC, there is no mitochondria. So in RBC, the glycolysis is only anaerobic in nature. So when there is anaerobic glycolysis in the RBC, this NADH, which is produced in the step number 4, is utilized for in the last step, where conversion of pyruvate is occurring to lactate. Here, this NADH is used and NAD is released for reutilization in the step number 4, so that glycolysis can continue in RBC. Okay? So, 2,3-BPZ, it is a negatively charged molecule because of the multiple phosphate group. 2 numbers of phosphate groups are there. It binds with the beta globin of the hemoglobin chain, because beta globin chain contains positively charged amino acids, so it binds there. So binding causes binding. Yes, stand up. What happened? Why you are laughing? How? Come this side. Tell me. So long what I have explained. Aja. Come. At least don't disturb me. Okay? Sit down. So, binding of 2,3-BPZ to the hemoglobin reduces the affinity for oxygen, affinity of oxygen towards hemoglobin. That means easily oxygen can be released from the hemoglobin. So, where we need it? When there is high altitude, there is partial pressure of the oxygen is very less. Then in obstructive airway disease, oxygen diffusion in the blood is very less from lung. Then anemia, oxygen carrying capacity has gone down. Cardiac failure, supply of the blood is in the periphery is very less. Blood transfusion, due to storage of long storage of blood in acid-saturated dextrose medium, acidic medium, destroys 2,3-BPZ. So to prevent that, to prevent destruction of 2,3-BPZ, we add one adenosine, inosine. This is your nucleoside. We add inosine so that this destruction of the 2,3-BPZ is prevented. Then hemoglobin F, there is increased 2,3-BPZ. There is in hemoglobin F, BPZ, 2,3-BPZ binding is less. So affinity of oxygen towards fetal hemoglobin is more. So that more amount of blood can be carried through placenta from mother's blood to the fetus. 2,3-BPZ, it shifts the oxygen dissociation curve to the right, means release of oxygen is favored. Now coming to the Pyruvate Dehydrogenase complex, now we have produced pyruvate in the glycolysis. That pyruvate has to undergo complete oxidation in the Tricarboxylic Acid cycle to give full oxidation of glucose. From the glucose, first part of reaction, that is glycolysis, pyruvate is produced. Now pyruvate will take part in the reactions of TCA cycle. But molecule which starts TCA cycle is acetyl-CoA. Now pyruvate has to change, or it has to synthesize acetyl-CoA to take part in the TCA cycle. Now, short note, metabolism of pyruvate or just pyruvate, they will give. Now, source of pyruvate from glycolysis, from lactate. Lactate pyruvate is converted to lactate in anaerobic glycolysis. It is reversible reaction. From lactate, again pyruvate can be synthesized by the lactate dehydrogenase itself. So that is also again another source of pyruvate. Then alanine is an amino acid, a non-essential amino acid. By transamination, this pyruvate is produced. Glycine, through conversion to serine, by serine gives pyruvate. Serine itself gives pyruvate by the action of serine dehydratase. Then cysteine by desulfatase enzyme, cysteine by acted upon by the enzyme desulfatase and gives pyruvate. These are the pyruvate. Now, where pyruvate is used? Pyruvate is used mainly in the tricarboxylic acid cycle. From pyruvate, there can be lactate formation through lactate dehydrogenase. through oxidative decarboxylation, acetyl-CoA can be produced, which can go to TCA cycle. Then pyruvate by pyruvate carboxylase enzyme can be converted to oxaloacetate. Then pyruvate can be utilized for synthesis of non-essential amino acids like alanine by transamination reaction. This is the metabolism. When pyruvate metabolism of pyruvate is given a short note, you have to give source and utilization. But this much only. So fate of pyruvate, what I have told. Pyruvate is produced in the cytosol because glycolysis is occurring in the cytosol. So that needs to be carried inside the mitochondria for further metabolism. But pyruvate can easily cross the outer mitochondrial membrane, but inner mitochondrial membrane it cannot cross. There it takes the help of hydrogen pyruvate transporter. Hydrogen pyruvate transporter. Through that transporter, it enters in the mitochondrial matrix, and with the help of PDH Pyruvate Dehydrogenase complex, it is converted to acetyl-CoA, and acetyl-CoA takes part in the TCA cycle in the mitochondrial matrix. So, yes. The PDH complex is given as short note, pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex, it is nothing but a multi-enzyme complex. It contains 3 enzymes. Pyruvate dehydrogenase complex itself, then dihydrolipoyl transacetylase. Dihydrolipoyl transacetylase or dihydrolipoyl acetyl transferase, same thing. And third enzyme is dihydrolipoyl dehydrogenase. So it contains 3 enzymes: pyruvate dehydrogenase, then dihydrolipoyl transacetylase. Number 3 is dihydrolipoyl dehydrogenase. In exam, don't write this short forms. Nah? So these 3 enzymes, they remain bound together and act upon the pyruvate. During the total process of conversion of pyruvate to acetyl-CoA, the substrate of these enzyme complex remain bound with the enzyme so that effective conversion is possible. Okay. This enzyme complex uses 5 numbers of coenzymes. They are three enzymes, 5 coenzymes. At TPP, then coenzyme A, then FAD, NAD, lipoamide. Number 1, 2, 3, 4 are the coenzymes of vitamin, and number 5th is the coenzyme of a vitamin-like substance. Actually, it is lipoic acid. Lipoamide is a coenzyme of lipoic acid. Okay. Next reaction is pyruvate converted into acetyl-CoA by use of 5 coenzymes and 3 enzymes and produce 1 NADH. Okay. Now this is the reaction. You have to draw this. Other PDH complex, when you answer, you have to draw this diagram. Okay. So this is the pyruvate. This is the thiamine pyrophosphate, the coenzyme of vitamin B1. So thiamine pyrophosphate take up the ethyl group from the pyruvate and becomes hydroxymethyl group, hydroxymethyl TPP, hydroxymethyl thyroid thiamine pyrophosphate. Then this hydroxymethyl pyrophosphate hydroxymethyl group is transferred to oxidized lipoamide and as acetyl. The acetyl group of this hydroxymethyl pyrophosphate, hydroxymethyl thiamine pyrophosphate is transferred to oxidized lipoamide and acetyl lipoamide is formed by the enzyme dihydrolipoyl acetyl transferase. This acetyl transfers the acetyl group from hydroxymethyl thiamine pyrophosphate to the oxidized lipoamide and it becomes acetyl lipoamide. This reaction is catalyzed by pyruvate dehydrogenase. This is by dihydrolipoyl transacetylase. Now the same enzyme dihydrolipoyl transacetylase transfers again acetyl group to the coenzyme to form acetyl-CoA. And this lipoamide, it becomes reduced lipoamide. Now to carry on this cycle, lipoamide need to be oxidized again. That is done by dihydrolipoyl dehydrogenase. FAD, flavin adenine dinucleotide, is reduced to FADH2 and reduced lipoamide is oxidized to oxidized lipoamide. Okay? Then afterwards, this FAD is regenerated and utilizing one NAD and NADH is produced. So here we are getting one reducing equivalent. It takes part in electron transport chain and gives 2.5 numbers of ATP. From one glucose molecule, we are getting 2 pyruvate, so total we are getting 5 ATP. Okay, from glucose here in this stage. So this is the link reaction between the glycolysis and TCA cycle. First part is called oxidative decarboxylation. This is carbon dioxide is removed. Then transfer of acetyl group in second reaction and regeneration of oxidized lipoamide. This is 3 steps reaction. No confusion, no? This is the regulation. So when there is increased NADH or increased acetyl-CoA, when there is increased acetyl-CoA, then it inhibits the pyruvate dehydrogenase. Substrate inhibition. That substrate causes inhibition of the pyruvate dehydrogenase. When there is increased NADH, that inhibits pyruvate dehydrogenase so that no more extra NADH is produced in the reaction. Pyruvate dehydrogenase is also regulated by covalent modification. Pyruvate dehydrogenase is active in dephosphorylated form. When it is phosphorylated, it becomes inactive. Okay? Now acetyl-CoA, NADH, ATP, these activate PDH kinase. PDH kinase is another enzyme which phosphorylates PDH. Kinase, whenever there is a kinase, it will phosphorylate. So PDH kinase means that enzyme which phosphorylates PDH. So this acetyl-CoA, NADH, ATP, it activates PDH kinase. As a result, PDH kinase will phosphorylate PDH and make it inactive. This is the way how acetyl-CoA, NADH, ATP, they inhibit PDH And coenzyme A, NAD, ADP. Presence of NAD, more NAD. Presence of ADP means there is lacking of ATP. Okay, so we need to increase NADH for ATP. So NAD, what it does? It inhibits PDH kinase. Inhibition of PDH kinase, what it does? It prevents phosphorylation of PDH. So PDH remains active. There is increased acetyl-CoA. More acetyl-CoA will take part in TCA cycle. More energy will be generated. This is the covalent modification, means regulation of PDH, pyruvate dehydrogenase complex by covalent modification. Okay. Come. So clinical aspects, these problems are not that much, but commonly if there is any clinical any disease which prevents this, there is deficiency of pyruvate dehydrogenase. It is the inherited PDH deficiency. It is characterized by lactic acidosis. Pyruvate cannot be converted into acetyl-CoA, so that is diverted to the lactate formation. So there will be lactic acidosis. Then neurological disturbance will be there, and this is the most common biochemical cause of congenital lactic acidosis. Though this condition is very rare. This is the rare disease, but usually when it occurs, it is caused by this inherited PDH deficiency. Okay. Now there are chemicals which inhibit this PDH, arsenite and mercuric ion. They causes inhibition of this coenzyme, thereby not making it available for the PDH complex to work. So indirectly it inhibits PDH complex. Dietary deficiency of thiamine, there will be deficiency of TPP, so PDH complex activity will be reduced. Okay, now this ends this part of this one slide. Then we will go to next part. Citric acid cycle, this is the metabolic cycle which essentially involves oxidation of acetyl-CoA to carbon dioxide, producing energy as ATP or reducing equivalents. So named due to the participation of tricarboxylic acid, that is citrate, C-aconitate, isocitrate. These are the tricarboxylic acids. From these, they have given the name tricarboxylic acid cycle. It accounts for about 60 to 70% of the total ATP generated in the body. Some amount of ATP are generated at substrate level, but TCA cycle contributes 60 to 70%. This is the major pathway of ATP generation, energy generation. This cycle utilizes about 2/3 of that total oxygen consumption in the body. This cycle is also known as Krebs cycle or citric acid cycle because citrate is taking part, or metabolic traffic circle. Metabolic traffic circle. This is the final common metabolic pathway for oxidation of carbohydrate, fatty acids and amino acids. Sir Hans Krebs in 1937 first described this cycle completely, and he was awarded Nobel Prize in the year 1953. The tricarboxylic acid, which is taking part in the condensation of oxaloacetate with acetyl-CoA, is later on identified as citric acid. That's why it is also called citric acid cycle. It occurs in the mitochondrial matrix. Okay? Only one enzyme of TCA cycle is bound to the mitochondrial membrane. This is succinate dehydrogenase. In the electron transport chain, the complex 2. Complex 1 is NADH dehydrogenase. Complex 2 is succinate dehydrogenase. This succinate dehydrogenase remains bound with the membrane. As we know, all the enzymes or complexes are bound to the inner mitochondrial membrane. All the complex of electron transport chain are bound to the inner mitochondrial membrane. So, being one of the complex, that means complex 2, succinate dehydrogenase, which catalyzes succinate to fumarate, remains bound in the mitochondrial membrane, inner mitochondrial membrane. Other all reactions are occurring in the mitochondrial matrix. NADH is produced, one NADH, one FADH2, one ATP is produced. Actually, ATP is produced not directly. First GTP is produced, and after that GTP is converted into ATP. Oxaloacetate acts as a catalyst. In the cycle, you can see the oxaloacetate, one of the component condensing with acetyl-CoA to produce citrate. And after the whole cycle, again oxaloacetate is regenerated. So, it is not synthesized, it is acting as a catalyst. It is taking part in the reaction, and after the cycle is complete, it is again regenerated. Okay? Synthesis of non-essential amino acid than heme, purine and pyrimidine occurs from the intermediates of TCA cycle. These are some considerations, important points about TCA cycle. Intermediates also act as regulatory factor in number of pathways. This is the schematic diagram of the cycle. So, we will use mnemonics to remember this. Cricket coach of India appoints smart staff for main office. Okay? So, this will give you the clue to remember intermediates of TCA cycle. Wherever there is N, that will be site of NADH generation. Wherever there is capital A, there will be site of GTP generation, that means ATP generation. Okay? So, in this pathway, the acetyl-CoA and oxaloacetate, they combine to form citrate. This is a 4-carbon compound, this is a 2-carbon compound. So, they will produce a 6-carbon compound. 6-carbon compound, citrate. The enzyme is citrate synthase. This is not it is not clear. Citrate synthase. First enzyme is, this is a 8 citrate synthase. Now citrate synthase will produce citrate, which is a 6 carbon compound. From citrate, alpha-ketoglutarate will be synthesized by aconitase. This is a two-step reaction. In between, isocitrate is formed. So, in the mnemonics, in isocitrate, we have N. So, when isocitrate is converted into alpha-ketoglutarate, it will produce a NADH. One molecule of carbon dioxide will be released. This is the first decarboxylation in the cycle. Okay? The enzyme is isocitrate dehydrogenase. From citrate to isocitrate, the enzyme is aconitase. Aconitase. Next is isocitrate dehydrogenase. Isocitrate dehydrogenase. Then isocitrate dehydrogenase is converted into alpha-ketoglutarate. Then alpha-ketoglutarate is converted into succinyl-CoA. The enzyme is alpha-ketoglutarate dehydrogenase. It is like that of pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate dehydrogenase. In case of pyruvate dehydrogenase, we have seen there are 3 enzymes. Here also there are 3 enzymes. Alpha-ketoglutarate dehydrogenase, alpha-ketoglutarate dehydrogenase, dihydrolipoyl succinyl transferase, alpha-ketoglutarate dehydrogenase itself, then dihydrolipoyl succinyl transferase, then dihydrolipoyl dehydrogenase. Dihydrolipoyl dehydrogenase. So these are the 3 enzymes for alpha-ketoglutarate dehydrogenase. They give alpha-ketoglutarate dehydrogenase as short note. This enzyme also, this enzyme multi-enzyme complex also, it uses 5 coenzymes. The same coenzyme that is used in the pyruvate dehydrogenase. TPP, coenzyme A, FAD, then NADA, then lipoamide. So first reaction, it is irreversible and it is the regulatory enzyme. The coenzyme which is released here, then coenzyme, the acetyl-CoA condensing with oxaloacetate, releasing coenzyme. That this coenzyme is again utilizes in the reaction number 4, catalyzed by alpha-ketoglutarate dehydrogenase to produce succinyl-CoA. This is the second reaction, step 2. Citrate is converted into isocitrate with intermediary reaction producing cis-aconitate. Then isocitrate is converted into succinyl alpha-ketoglutarate. In between, one molecule is transiently produced. This is oxaloacetate. Here again, decarboxylation occurs. This is the second. This is the first decarboxylation. First oxidative decarboxylation. Now, one thing I'll tell you, the carbon dioxide released in the reaction is not the carbon dioxide what is present, is not the carbon which is present in acetyl-CoA. It is some other carbon. It is from the oxaloacetate. The carbon is released as carbon dioxide. Succinyl-CoA is formed by the enzyme alpha-ketoglutarate dehydrogenase. One NADH is produced and second molecule of carbon dioxide is released. And this is the second oxidative decarboxylation. So we started with 6 carbon, 2 carbon from the acetyl-CoA, 4 carbon from the oxaloacetate, and we have released 2 carbon dioxide, and that means 2 carbon. So the resultant succinyl-CoA is 4 carbon compound. This is also irreversible step and regulatory step. The enzymes which are in the multi-enzyme complex of alpha-ketoglutarate dehydrogenase are alpha-ketoglutarate dehydrogenase, dihydrolipoyl succinyl transferase, and dihydrolipoyl dehydrogenase. Reaction is same. Factors, 5 factors: TPP, lipoamide, then FAD, NAD, and coenzyme. This will come afterwards. Then succinyl-CoA, with the help of succinate thiokinase, forms succinate. Here, a substrate level phosphorylation occurs and GTP is produced. GTP. Afterwards, this GTP is converted into ATP. Yeah, GTP can donate its terminal phosphoryl group to ADP to form ATP. Liver and kidney, these are where the gluconeogenesis occurs, contains 2 isoenzymes of succinate thiokinase, one for ADP, another for GTP. So there are 2 forms of succinate thiokinase. One form uses GDP as substrate to produce GTP, another directly it can produce ATP using ADP. Formation of fumarate, then succinate with the help of succinate dehydrogenase forms fumarate, one FADH2 is produced, and this enzyme remains bound with the inner mitochondrial membrane as complex 2 of electron transport chain. Then formation of malate by fumarase. Fumarase acts upon the fumarate and produce malate. Then malate with the help of malate dehydrogenase produce again oxaloacetate. It is actually oxaloacetate is not synthesized, it is recovered from the reaction. So it acts as catalyst. So we can use the mnemonics. What are the intermediates? These are citrate, then isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, and malate, then oxaloacetate. Energetics, wherever there was N, there will be NAD production. So there will be 3 one NAD will be produced in the reaction catalyzed by isocitrate dehydrogenase. One will be produced in the reaction catalyzed by the alpha-ketoglutarate dehydrogenase. Another will be produced in the reaction catalyzed by malate dehydrogenase. So 3 NADH is produced in the TCA cycle, 1 FAD where catalysis occurs by succinyl dehydrogenase, then one substitutable phosphorylation occurring in the succinate thiokinase reaction. So we have 3 ATP and 1 FAD, H2 and 1 ATP. Regulation of Krebs cycle. ATP NADH, they inhibit citrate synthase, isolated dehydrogenase and alpha-ketoglutarate dehydrogenase. And ADP, NAD, they activate isolated dehydrogenase and alpha-ketoglutarate dehydrogenase. So, ADP, when it is more, it activates TCA cycle. When ATP is more, it inhibits TCA cycle. Feedback inhibition, citrate synthase is inhibited by citrate, isolated dehydrogenase is inhibited by isocitrate, alpha-ketoglutarate, then alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA. These are the feedback inhibition. Regulation by calcium. Calcium activates isocitrate dehydrogenase and alpha-ketoglutarate and enhances TCA cycle occurrence of TCA cycle, because when there is muscle contraction, the body needs more ATP, so release of calcium ion, it activates enzymes of TCA cycle and thereby increasing ATP production. Inhibitors of Krebs cycle. These are the chemicals that inhibit Krebs cycle. These are fluoroacetate, arsenite and malonate. Fluoroacetate, this will inhibit aconitase. Fluoroacetate will inhibit aconitase, arsenite will inhibit alpha-ketoglutarate and malonate will inhibit succinyl-CoA. Okay, so just to remember, you can use this FAMAKS. Okay, this can be taken as mnemonics. TCA cycle, it is the final common metabolic pathway. In the first stage, carbohydrate is converted into simple sugar, glucose, lipid into fatty acid, protein into amino acids. In the second stage of metabolism, that means intermediary metabolism, this acetyl-CoA is synthesized from glucose or fatty acid or amino acids. In stage 3, acetyl-CoA is completely oxidized into carbon dioxide and reducing equivalents are produced along with the production of substrate level ATP. Reducing equivalent, they take part in electron transport chain and produce ATP. These are the amino acids which can enter in TCA cycle to get metabolized. Their carbon skeleton is metabolized in the TCA cycle. Glycine, alanine, cysteine, serine, threonine and tryptophan, their carbon skeleton is converted into pyruvate and then take part in the TCA cycle. Phenylalanine, isoleucine, tyrosine, tryptophan, leucine, they form acetyl-CoA. Leucine is purely ketogenic. It is hydrolyzed into acetyl-CoA and can take part in the TCA cycle. Here you see from acetyl-CoA, pyruvate to acetyl-CoA is catalyzed by pyruvate dehydrogenase complex. But acetyl-CoA cannot form pyruvate because of the PDH complex is completely irreversible state. That's why acetyl-CoA cannot be converted into pyruvate. So it cannot synthesize glucose. So aspartic acid, it can form oxaloacetate, phenylalanine tyrosine. It takes part in, it can be converted into fumaric with their carbon skeleton. Then valine, isoleucine, methionine, they form succinyl-CoA, glutamate, arginine, histidine, they form alpha-ketoglutarate and ultimately metabolize in the TCA cycle. But they are not the primary substrate for energy production. Energy production is primarily from the glucose or lipid, fatty acid. Short note, TCA cycle is a amphibolic pathway. Explain. Metabolic pathway is called amphibolic pathway when it has both anabolic and catabolic role. So long we have described, they are the catabolic component of TCA cycle. Acetyl-CoA is catabolized completely into carbon dioxide in TCA cycle. This is the catabolic pathway. But anabolic pathway is what? Citrate. This is the citrate in mitochondrial matrix. It is carried to the cytosol and acted upon by citrate lyase. Citrate lyase into citrate into acetyl-CoA and oxaloacetate. So oxaloacetate. These acetyl-CoA then take part in formation of fatty acid, synthesis of fatty acid. This is the one anabolic pathway that is performed by the TCA cycle. So this acetyl-CoA, it takes part in synthesis of fatty acid, it takes part in synthesis of cholesterol. Then alpha-ketoglutarate, it forms the glutamic acid, amino acid. From glutamic acid, gamma amino, benzoic acid. This is a neurotransmitter. It is synthesized from the alpha-ketoglutarate. Then succinyl-CoA, it is used for heme synthesis. Succinyl-CoA plus glycine is condensed. The first step of heme synthesis. Okay. Then succinyl-CoA, it transfers coenzyme group in the ketolysis reaction. Acetoacetate is converted into acetoacetyl-CoA, taking the coenzyme group from succinyl coenzyme. Succinyl coenzyme. Okay. So oxaloacetate, again, it can be converted into glucose. It can be converted into non-essential amino acid aspartate, which is again utilized for synthesis of non-essential amino acid, purine, pyrimidines. All these things, they are synthesized from the intermediates of TCA cycle. So these are the anabolic component of TCA cycle. That's why it is called amphibolic pathway. Anaplerotic reaction. You have seen the major 60 to 70% of ATP is generated in the body from the TCA cycle. So if any component from the TCA cycle is withdrawn or it goes into depletion, the TCA cycle will be hampered and there will be disturbance in ATP generation. Okay. So there are almost many of the states wherever there is, in almost all states, there are cycles which can replenish the deficiency of intermediates of TCA cycle. Okay, these reactions which replenish the intermediates of TCA cycle is called anaplerotic reactions. Okay, so aspartate by transamination it gives oxaloacetate. Pyruvate also can be converted into oxaloacetate. Glutamate by transamination and deamination it gives alpha-ketoglutarate. Valine, leucine, isoleucine, methionine, and propionyl-CoA. Propionyl-CoA is produced from oxidation of or-chain fatty acid. Okay, or-chain fatty acid, then it can be converted into succinyl-CoA. So these molecules, they replenish succinyl-CoA when required. Then glucosonic amino acid, they can provide fumarate. Then pyruvate with the help of malic enzyme is converted into malate. Okay, so these are the anaplerotic reactions or anaplerosis. So when anaplerotic reaction, short notes is given, so you have to give this reaction in not in detail, just from which substrate what is produced and how it is replenishing. Okay. So we are using 5 coenzymes in alpha-ketoglutarate dehydrogenase. So these are 4 vitamins that are required to TCA cycle for functioning, and 1 vitamin-like compound lipoic acid. Another, explain why. Fat is burned on the wick or flame of carbohydrate. See, from the fat, that means triacylglycerol, free fatty acid is produced. Free fatty acid is produced, fatty acid undergoes beta oxidation to produce acetyl-CoA. Now, to undergo further metabolism, the oxaloacetate is required. Acetyl-CoA condenses with oxaloacetate to form citrate. Oxaloacetate is nothing but carbohydrate. So, to burn the fat, we need the carbohydrate. Without oxaloacetate, this acetyl-CoA cannot enter in TCA cycle. Understand? Clear? So, oxaloacetate is required. That's why we tell fat is burned on the wings of or flame of carbohydrate. TCA cycle is operative only under aerobic condition. Have you seen anywhere oxygen is required? No. Nowhere in cycle there is oxygen we utilize. But to be operated, it needs NAD, FAD. Okay? These NAD and FAD, they need to be oxidized in electron transport chain. The electron transport chain requires oxygen. Essentially, it requires oxygen. Without oxygen, electron transport chain cannot occur. Okay, ultimate acceptor of reducing equivalent is the oxygen. Okay, that's why no electron transport chain, no TCA cycle. So it can occur only in aerobic condition. Okay? If you read today, you will remember. These are the key points of TCA cycle. Primary purpose is to oxidize acetyl-CoA to carbon dioxide and generate reduced coenzyme NADH, FADH2, ATP. Steps: cycle begins with acetyl-CoA, combines with oxaloacetate to form citrate. Through a series of 8 reactions, 2 carbons are released as carbon dioxide and oxaloacetate is regenerated. Location occurs in mitochondrial matrix. Regulation control at 3 main points: citrate synthase, isolated dehydrogenase, alpha-ketoglutarate dehydrogenase. Key intermediates: citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. remember this chart you will be able to prepare. Okay? Anabolic in nature, while the cycle does not use aerobic in nature, while the cycle does not use oxygen directly, it requires aerobic condition because it relies on NAD and FADA generation from the electron transport chain which needs oxygen. Biological significance, beyond energy production, the TCA cycle is amphibolic, meaning it functions in both catabolism and anabolism. Okay? So, we have seen so many pathways that opens to the TCA cycle, so many pathways that goes from TCA cycle to synthesize fatty acid, cholesterol, non-essential amino acid, purines, pyrimidines, all these things. Okay? So, it is rightly said that TCA cycle is a metabolic traffic circle. TCA cycle is called metabolic traffic cycle. Explain why. Okay? These are the some short notes and explain why. That is given from TCA cycle. Of course, long answer type of question can be possible. Write the steps of TCA cycle, then sites of reducing equivalent and ATP generation, and regulation of TCA cycle. So, in these three parts, it can be given as a long question, others, these are the short questions. So, thank you very much. Read TCA cycle, very important. Afterwards, you will have all the cycles related to TCA cycle because it involves all the food particles. It is metabolized through TCA cycle only. Okay?